Today I'll be talking about a project entitled The Genetic Determinants of mRNA Degradation Rates in Mammals. So just to start off with a little bit of background introduction, many of us are familiar with this life cycle of mRNA since high school biology. It's a very beautiful process, and there are many different layers of regulation at every single one of these individual steps. And it's really defined the last 13 years of what I've been interested in studying. So of course, RNA is born through the process of transcription, and subsequent to that, it's processed through the removal of introns and the addition of the poly A tail. And eventually it's exported to the cytosol, whereby at times it might be localized or translated into proteins. And the final step of this process is the degradation or death of RNA. So over the past 6 years or so, I've taken like a detour in my life to try to understand the determinants of gene expression levels and how the genome encodes the transcription rates of RNA. And we published several papers in that regard. And just last year, we also published a paper looking at single cell sequencing data to try to evaluate different events of alternative polyadenylation across early development of a mouse embryo. But today I'll be talking about my most recent return to my origins, which is the study of post-transcriptional gene regulation, which I've been investigating for the past 2 years, and we've released this work on bioRxiv recently. So first of all, if we think about mRNA metabolism and its role in disease, there are many different components of the system that can break. So for example, reduced RNA binding protein expression, the aggregation of RNA binding proteins, or their sequestration to repeats can all lead to alterations of RNA metabolism, including changes in splicing, adenylation, and transcription, mRNA transport, its translation, or decay. And I'll just be focusing on one subproblem of this, looking at mRNA decay. So to tie this in a little bit in disease, there's some reviews that have actually looked at different mechanisms of RNA decay that could lead to disease. So for example, it's been discovered that deletions in degradation elements in c-Myc, COX-1 and 2, and CCND1 have led to outcomes such as myeloma, T-cell leukemia, and lymphoma. Mutation and degradation elements in TNF-alpha have been shown to play a role in the emergence of inflammatory diseases, such as chronic inflammatory arthritis or bowel disorders. Stop codon mutations can lead to nonsense-mediated decay, leading to the degradation of alpha globin, and that's led to alpha thalassemia, leading to low hemoglobin levels in the blood. It's also been discovered that increased stability of the transferrin receptor has led to reduced ferritin synthesis, which might predispose an individual to the progression of Alzheimer's disease. And of course, many of us in the genetics community are really interested in these genome-wide association studies, which over time have accumulated and discovered more and more single nucleotide polymorphisms in our genome, which might play a role in disease. One of the complex areas of interpreting GWAS analyses is that they often lead to ambiguity in determining the causal SNP involved because of the property of linkage disequilibrium. There are many different regions that are statistically associated with a change in gene expression. So there are certain critical challenges in interpreting GWAS and linking genotype to phenotype. We'd like to be able to precisely identify the causal variants within an associated locus and determine the mechanisms by which these variants result in the observed phenotypes. It's also really imperative to be able to find the associations that lead to modified gene regulation, whereby whether it be which gene or genes are affected, in which cell types or situations gene expression is modified, or which upstream regulators are perturbed. There was this really interesting study from Jonathan Pritchard's group in 2016, which enumerated the different types of gene regulation and how they might play a role in governing final protein abundance. And one of his findings was that approximately 80% of regulatory quantitative trait loci actually play a role in transcriptional gene regulation, whereas there's an additional 10% of QTLs that might actually disrupt the degradation rate of RNA. And that's what I'll be focusing my talk on today. So just to switch gears for a moment and think a little bit about RNA therapeutics, all of our lives have been impacted in the past few years by COVID-19. But one of the beautiful things in the past few years is this proof of principle demonstration that mRNA vaccines are very effective. And so just to briefly walk through how they work, the virus has a coat protein or a spike protein. And if we encode that spike protein in RNA and encapsulate it in lipid nanoparticles, you can deliver this payload into human cells, whereby they're translated and present themselves as antigens to the immune system to develop a robust immune response. Now, there's several critical limitations still with these types of vaccines that we'd like to address. And one of them is encapsulated by this quote from a review article. And it says, following injection into muscles, synthetically made mRNA likely undergoes rapid RNA degradation by both extracellular and intracellular RNases. Since the half-life of mRNA in the cytoplasm is directly associated with protein expression, it is critical to maximize the stability of RNA. Just to talk a little bit about the distribution of vaccines around the world, there's also tremendous inequity in where those vaccines have been distributed, in that a lot of the Western hemisphere and Western countries have been able to successfully distribute the vaccine, whereas developing nations in Africa and Asia have less access. So you can imagine that one of the limitations in this problem is that if less RNA was needed in each individual, if we were able to develop a therapy that was more stable, we would then be able to distribute this more cheaply to developing nations. So I've talked about two different applications, mRNA vaccines, which in the future has implications for COVID, the flu, Zika virus, HIV, cancer, and other infectious diseases. And I've also talked about genetic diagnostics and the interpretation of eQTLs and GWAS analyses. And I'd like to develop a bridge that connects these two worlds. And of course, there are several bridges, but the one that I'm focusing on is the bridge of post-transcriptional gene regulation and the fundamental biology of what's governing mRNA turnover. My journey into this problem began about 10 years ago when there was a grad student next to me named Noah. And he was really interested in that time to be able to predict mRNA half-lives from mRNA sequence information. So his approach to this question was that he collected a number of different mRNA sequence features, and he trained a Mars nonlinear regression method to try to predict half-life. Now, the best R squared he could achieve was an R squared of 0.39. And to delve into this model a little bit, we could see the different features going into the model that was most highly predictive of half-life. The number one feature in his model is ORF exon junction density, or the number of exon junctions per kilobase of an open reading frame. The single feature is highly correlated to mRNA half-life. In addition, if he adds two additional features, that of 3' UTR length or the presence of familial 2 binding sites, you can get incrementally better. But after that, it seems a little bit hopeless because if you add many, many more features, the model kind of tapers out in its ability to do any better. So that was kind of the state of the art in this question. And the question had kind of been abandoned for many years, or at least there was not subsequent work that really improved upon this. So I wanted to kind of revisit this problem and see if we could do any better. So the first key question I wanted to address to tackle this problem is I wanted to ask, can we derive a more accurate measurement of RNA half-life through a meta-analysis of experiments? And my goal here was to systematically collect mRNA half-lives across different studies and cell types. My next question is, can we directly predict mRNA half-life from mRNA sequence? And then my goal here was to derive a better machine learning or deep learning model to predict stability. And my third goal was to ask, can we predict the functional consequences of mutation on RNA stability? And my goal here was to benchmark how these types of models can predict mutational effects. Now, to address these questions, we first need to ask how half-life is measured to begin with. So there's a number of different techniques, but largely they break down into two different methods. In method one entails the post-labeling of nascent RNA with a modified nucleotide, such as 4-thiou or bromouridine. So, and subsequent to this, you can measure the concentrations of old versus newly synthesized mRNA over time. Method two entails a transcriptional shutoff experiment with a drug, such as actinomycin D, followed by monitoring of RNA levels over time. And so just to give you three examples on the top right here, we can plot the abundance of RNA as a function of time and fit an exponential decay curve to extract the coefficient of half-life, which merely indicates the amount of time expired before half of the initial concentration of the RNA still remains. So my first aspect of the meta-analysis was that I embarked on a relatively painful journey to mine the entire literature and try to get my hands on every half-life data set that ever existed, as far as I could tell, in mammals. So I synthesized a number of different experiments that were generated out there, and they encompass many different cell types. I generated a table of the references, the species, whether they be human or mouse, the method of origin used to measure those half-lives, and the groups of origin for those. And all of these were developed on different gene sets, so I needed to standardize them all into one matrix with a shared gene set. I also did this in the mouse, and that encompassed numerous different methodologies and groups and cell types in the mouse as well. So the first question I wanted to ask is, how correlated are different samples from different groups to establish whether this is a reproducible measurement to begin with? So the first analysis I performed was looking at the pairwise Spearman correlations between every sample to each other sample. And I got a heat map that looks something like this, whereby we can see in the top right-hand quadrant of this heat map, a large number of studies from many heterogeneous groups achieve a decent pairwise Spearman correlation of about 0.6 or greater. Really, there's a single study from one group, Gitchman et al., that was a massive outlier relative to all other half-life data sets. So it is parsimonious to think that given that there are 30 other groups with somewhat concordant results, that this was more of an outlier. And then there's a group of studies that's kind of somewhere in between that has a weaker but still significant correlation to the other half-life data sets. Now I visualized this data a slightly different way by performing principal components analysis. And I colored each of these points according to the methodology of origin. I labeled them according to the group of origin, and the group of origin is colored according to the cell type of origin for these studies. Now, one would hope to see in a perfect world that different groups measuring half-lives in the same cell type would have similar clustering along this principal components analysis space or PC space. But that seems not to be the case. We can clearly see examples whereby in K562 cells, for example, we see some samples that are pretty far from other samples in this space. And that holds true for a number of different cell types. So that really is starting to suggest that there are a lot of prevalent batch effects in these types of data sets that we'd like to clean up a little bit. Another thing that I observed is that principal component 2 could actually strongly segregate the methodology of origin. So if a group used a pulse labeling based strategy versus a transcriptional shutoff based strategy, PC2 can actually segregate this type of information. So the methodology is a significant bias in these data sets, suffice it to say. Now, I want to develop a simple procedure to try to clean up some of these technical biases and batch effects. And so one of the things I did is just turn to literature and look at what people had been doing in the past. And there was something really interesting that popped out to me that just over 10 years ago, one of the first techniques that measured half-lives in both the human and mouse detected this really interesting evolutionary signature that if you look at the one-to-one orthologs between a human and a mouse, you can actually see pretty striking concordance in half-lives between these species. They achieved a Pearson correlation of 0.61. And so the procedure that I developed is that first I used my matrix of half-lives for each gene by each sample. And I did that separately for a human and mouse. I computed the first principal component of these matrices and get a single metric, just a simplified measurement of a cell type and method independent measurement of half-life for each gene. Then again, I asked this question again, could I gather the one-to-one orthologs between these two species and compare the relationship of half-lives? Doing that exercise, I got the following result. I was able to see a significantly stronger correlation between half-lives across these two species looking at one-to-one orthologs, indicating that by cleaning up some of this noise in the assays and batch effects, we can actually see this evolutionary signature that there's a much stronger conservation of half-lives than had been previously appreciated. And with these half-lives in hand, what I next wanted to do was to frame this as a machine learning or regression problem. My goal being to identify a set of features that might possibly influence mRNA half-life based upon a survey of the literature. So to achieve this problem, I used tenfold cross-validation to quantify how different subsets of the features impact model performance. And my goal was to use lasso regression to automatically learn which subset of features was maximally predictive while using the smallest set of features. Now, to walk you through a few of the types of features I used in my preliminary models, I used a technique called DeepRipe, which was published a few years ago, whose goal is to feed in an RNA sequence and make a prediction as to the likelihood that that sequence is bound to a number of different RNA binding proteins. So they trained this deep learning model using ClIP information, ClIP-seq information, which measures the binding of RNA binding proteins on mRNA. And so using their pre-trained models, I could then just feed in segments of the RNA and then ask it the likelihood that these sequences are bound to RNA binding proteins. Another area of interest that the literature has been developing over the past few years is this notion that codon composition actually might impact mRNA decay. It was a phenomenon described first in yeast and then later shown to exist in vertebrate organisms as well. So they derived a very simple metric. So basically for each mRNA, you can compute the frequency of each codon of each of the 61 nonstop codons that exist, and then look at the correlation between those frequencies and mRNA half-life. And by doing that simple exercise, you can then make a ranking of each codon according to the likelihood that that frequency is associated with increased or decreased half-life. So codon frequency is another type of feature that I was interested in using in my model. So to give you an overview of all the different types of features I considered, first I enumerated a set of basic features of an mRNA, including the GC content and length of the 5' UTR, ORF, 3' UTR, and intron length. And I also looked at ORF exon junction density. So this comprised only 8 features. Next, I enumerated all possible different k-mers that exist in a 5' UTR, and that from lengths 1 through 7 of all possible k-mers. That resulted in 21,844 features. You can do that same thing for the open reading frame or the 3' UTR. In addition to that, I computed codon frequencies in the open reading frame, leading to 61 features. Next, I looked at microRNA target predictions for microRNAs conserved among the mammalian lineage, and that corresponded to 319 microRNAs and their predictions along the transcriptome. And finally, I used 2 of these different deep learning engines to try to predict the likelihood that these mRNAs are bound by different RNA binding proteins. I used seqweaver and DeepRipe, resulting in 780 and 177 features, respectively. So these are the features that I was initially considering in the model. And now I'm showing you the results of the tenfold cross-validation results for each of the held out folds of the data. So I start off with the simplest model of them all. I just used 8 basic mRNA features. And that model in itself does pretty well. It gets a Pearson correlation of about 0.57 or so. Next, I integrated all possible 5' UTR k-mers from length 1 through 7, and it turned out it led to absolutely no improvement in the model. But then I looked at successive other types of features. So I looked at ORF k-mers from length 1 through 7, 3' UTR k-mers, or codon frequencies. And we could see that each of these did improve performance, but the very best performance arises just by using codons. Now I looked at additional, more complex models. So by combining codon frequencies with k-mers in the open reading frame, I actually saw that open reading frame k-mers led to no improvement in the model over just using codons alone. So I'm talking about this model relative to this model. And next I integrated microRNA binding predictions with 3' UTR k-mers, and I did actually see that that seemed to improve prediction relative to just using 3' UTR k-mers alone. Finally, if I combine codons, 3' UTR k-mers, and microRNA binding sites, I saw the biggest increase in performance. Next, I tried two more successive models, ones that incorporated these predictions of the RNA binding proteins along sequences. And I found a single model, one that incorporated seq-weaver predictions, microRNA binding sites, 3' UTR k-mers, codon frequencies, and the basic set of features that did the best of all. So summarizing all this information across the 10 folds of information, I was able to achieve a Pearson correlation of 0.68 with this lasso regression model. So what has this model learned? So to address that, I just ranked the different coefficients in this model. And the number one coefficient is the ORF exon junction density, which is a feature that was discovered long ago by Noah and others before me. But in addition, we saw that there are a number of different features corresponding to codon composition that are more frequently associated with faster RNA degradation than RNA stability. In addition, there were several other interesting k-mers that automatically popped out of this model. So one of the k-mers was UAUUUAU, which turns out to be the classic AU-rich element that's present in 3' UTRs. I also automatically discovered UGUAAU or UGUAUA, which turns out to correspond to the familial element. Finally, I also found AAUAA, which corresponds to the cleavage and polyadenylation element. So it was really nice to see among an unbiased search of over 60,000 different features that these came out among the top ranked features. Next, I also found different predictions from seq-weaver coming to the top, including the binding of RBM22 and SRF-SF3 and HNRNPUL1. And these are potentially novel factors, but it's really just generating a hypothesis that they may play a role in degrading RNA. But less is known about these factors and their role in mRNA decay. So that's where I could get with these lasso regression models. And next I wanted to ask, can we do even better than these linear regression models? So then I turned back to work that David Kelly and I have been pursuing over the past 6 or 7 years now, in which we've had some degree of success in training deep learning models in the context of regulatory genomics. David Kelly has formulated one problem in which he takes a large region of the human genome and trains a convolutional neural network to ultimately try to predict in a tiled fashion across a locus the likelihood that that region, the signal of ChIP-seq of different binding of transcription factors, cage information indicating transcriptional activity, or DNase hypersensitivity information indicating chromatin accessibility. He trained a multitask model to try to predict all of these in numerous different cell types simultaneously. Independent to his research, I was training deep learning models to feed in a promoter sequence and then attempt to integrate that information with features relevant to mRNA half-life to predict the steady state levels of mRNA as measured by RNA-seq. And finally, we put our efforts together when I joined Calico and worked with David to develop the Informer, which represents a collaboration between Calico and DeepMind to incorporate the ability to learn information about distal regulatory elements in predicting gene expression levels. So inspired by this lineage of work, we're hoping that this type of model might actually benefit the predictive task of predicting mRNA half-life. So towards that goal, we ended up training another deep learning, deep neural network to predict half-life from sequence. So the input to the sequence is basically a full length of an mRNA encompassing the 5' UTR or 3' UTR and a padding at the 3' end. So we can then take the sequence and one-hot encode it into a matrix represented by the 4 different nucleic acids. We also incorporated 2 additional pieces of information. So one information track that we incorporated is the position of the first reading frame. So it can have some sort of positional notion of the existence of an open reading frame. We also incorporated a last track indicating the presence of 5' splice sites. So if there's an exon junction, we're just annotating that that junction exists. So that's the input to the model, and we feed it through a number of different convolutional layers, again, as we trained in our previous models. But one different layer that we didn't use in our models is we used this recurrent neural network type of layer called a GRU, or gated recurrent unit. And the goal of a GRU is that it deals with the problem that mRNAs, of course, are of heterogeneous size. So you need some way to march along that sequence and summarize that information to create one consensus readout to summarize information along the sequence. So we can feed the output of that into a fully connected layer and then ultimately predict mRNA half-lives. We call this model Saluki. It's a hybrid convolutional and recurrent neural network model. And we call it Saluki because it's a hound dog trained to sniff out the function of genetic variants in RNA half-life. And because David loves dogs as well. So how does Saluki do in different contexts? So we benchmarked the performance of Saluki in both human and mouse independently relative to our best lasso regression models. And we actually found something really nice, which is that there's a very large increase in the performance when training this deep learning model relative to the very best lasso regression model I could possibly train. And we see that phenomenon to be true in both human and mouse. So to summarize that information, now we can see that Saluki was able to achieve a Pearson correlation of 0.77 between the observed half-lives and our predictions. Now, a lot of skeptics exist in the community when they look at a deep learning model and they ask, what could this 100,000 parameter model possibly have learned? So they frequently treat a deep learning model as a black box, and rightly so, because it's a pretty complex model to interpret. I actually started off as a wet lab experimentalist, so some of that mindset stuck with me in creating a black box, kind of like how an experimentalist treats a cell. In the wet lab, you look at a cell, it's a very complex machine, and you perturb it, and then you see what happens on the other end of that. And so you can kind of think of that about a neural network in the same framework. You could perturb a neural network with a different input, see what happens in its prediction, and hopefully learn something about what the model itself has learned. So our idea here now is to perform classic biology experiments, which many people do in the wet lab, except instead of in vivo, we're running them in silico on the computer. So one of these experiments that I performed is I performed an in silico mutagenesis on the entire transcriptome. So that is, I took each nucleotide of the 5' UTR ORF and 3' UTR of every gene, and then I mutated that into every other nucleotide and predicted what would happen as a consequence. And through that exercise, I was hoping to quantify which motifs might be enriched in this predictive engine. The second experiment that I did was an insertional analysis of motifs along the transcriptome. So for each of the motifs that I discovered, I inserted them at different positions along an mRNA for all the mRNAs in the transcriptome, and then measured on average how would they change RNA stability as a function of their position. And the third experiment I performed was a deletional analysis and saturation mutagenesis on a particular locus of interest that had been performed by an experimentalist in the past. And I was hoping to test the concordance of our predictions in silico with those measured in vivo. So if we actually look at the motifs that were discovered through this in silico uh, mutagenesis that I had performed, we can see several really interesting motifs pop out. So one of them turned out to be this element called the M6A element. This is methyl 6-adenosine. And this is a pathway that a lot of people have been working on for the past few years and has been implicated directly in modulating RNA half-life through the incorporation of a modified nucleotide in RNA. And this element is one that an M6A writer in the nucleus of the cell actually recognizes to deposit this mark. In addition to this, we were able to also detect the familial element, as you can see here, in all three functional regions. The AU-rich element, in contrast, we could detect in the 3' UTR. We also found a AU-rich element that corresponded to something kind of like the ELAV L1 and 2 element, or HUR, if you're familiar with that RNA binding protein. We found an element that looks kind of like a poly-A binding protein element, a poly-A element. We also found AAU-AAA, which is the polyadenylation element. And we see a number of different shorter elements that appear to be something that look like codons that were enriched in the open reading frame. So with these types of elements in hand, I tried to do an insertional experiment in which I inserted each of these different motifs at different bins across any mRNA in the transcriptome. And by averaging that information for each bin in the transcriptome, I could get kind of an average portrait of how this perturbation or insertion of this motif might impact RNA half-life. And from that exercise, we saw some really interesting patterns. We could see, for example, by inserting a splice site, it seems to have the greatest impact when it's closer to the 3' end of the open reading frame. So we think we're learning a lot of interesting positional dependencies that are somewhat novel, but at the end of the day, it would be really nice to test these inferences experimentally. In addition, we've also seen the integration of an AAU-rich AAU-AAA, the polyadenylation element, seems to lead to greater stability of the RNA when incorporated downstream in the 3' UTR. Whereas in contrast, if we incorporate a familial binding element or the AU-rich element here, it leads to decay, and that's dependent again on the position of the insertion. Finally, I did the same type of analysis of these insertional experiments, except now instead of motifs, I'm just using codons inserted along the open reading frame. And again, we saw some really interesting patterns whereby towards the beginning of an open reading frame, we see that there's either a very sharp drop-off in that there's enhanced RNA degradation associated with the presence of this motif, or in some cases, greater stability of the RNA depending on which of these motifs we're incorporating. And again, there are very strong positional dependencies here that are often related to the boundaries of the open reading frame. So just to show that this is actually a property that the deep learning model learned itself, but it's actually consistent with the literature, which has actually measured what they call the mean codon stability coefficient. And so that kind of alludes to the data I showed you earlier, that if you measure the frequencies of the codons, you can just look at the correlation with half-life. And by analyzing this deep learning model, I can compute the mean codon influence as predicted by this model. And we can actually see a pretty strong correlation between these codons and those previously detected in literature, exhibiting a Pearson correlation of 0.68. Next, what I wanted to do was to perform an in silico mutagenesis experiment. And there was this beautiful work by David Earley's group at UCSF, where many years ago, he published this Nature Biotech paper where he analyzed a single 3' UTR of the gene CXCL2, and he performed a tiling massively parallel reporter assay to take tiles along this 3' UTR, incorporate mutations along those tiles, and then measure the amount of repression or de-repression from the perspective of mRNA stability as a function of those little tiles marched along this UTR. From this functional assay, he could pull out really interesting regulatory elements, which often coincided with regions that are conserved in this 3' UTR. So I wanted to ask whether I could perform the same type of MPRA experiment in silico and get a readout that was somewhat similar. And indeed, I could see some qualitative similarities between an in silico experiment and that done in vivo. I could see a few elements that lined up very well, although certainly there were some regions that lined up poorly and were perhaps false negatives of my technique. So the technique is not perfect, and some of these regions might be due to cell type specific elements as well, which is something that's missed by our current algorithm. Another interesting experiment he did in the study was to take one specific region of this UTR and then perform a saturation mutagenesis experiment in which he takes every nucleotide, wild type nucleotide of the sequence, mutates it into every other nucleotide, and then measures its impact with respect to mRNA stability. So I wanted to perform that exact same experiment, saturation mutagenesis experiment now in silico to see if we could achieve something similar. And strikingly, we could actually see something similar in that many of the same AU-rich elements that were lighting up with respect to the functional assay were the same ones our computational engine could predict as well. So to summarize all this information, I could plot the predicted variant effect for every one of these mutations on the x-axis and the observed variant effect on the y-axis as measured by the MPRA, and see that they agree somewhat well with a Pearson correlation of 0.65. So just to conclude a little bit here, I've generated a large compendium of human and mouse half-life datasets for the community. And hopefully this is a useful resource for the community moving forward as a database to compare future mRNA half-life studies to. I've identified a major source of bias in the measurements of half-lives and attempted to remove their impact. Next, I've developed a state-of-the-art sequence-based model that is 50% better than existing models in that it achieves an R² of 0.59 relative to the current state-of-the-art of 0.39. Finally, a model such as this one is nice in that it can be used to predict the functional consequences of mutation on mRNA half-life, and moreover, it can be deployed very rapidly on the entire transcriptome. So some of the future directions of this work, as I alluded to, it's very complex to be able to interpret genome-wide association studies. And in theory, it would be really nice if we could take the subset of mutations that intersect those of the exons of an mRNA, and then be able to use our engine to be able to predict the impact of those variants. And by doing that, it can at least inform us, give us a hypothesis as to a mutation that might be the causal mutation, and narrow down the focus into such subset of mutations that might be more deleterious. So this is the predicted causal SNP in this scenario, for instance. So where do we go from here? We would really like to be able to use these mutational scores to investigate disease genetics by improving fine mapping and generating mechanistic hypotheses, as I mentioned, about the function of variants, especially rare variants, which are notoriously difficult to interpret. And in the future, we've developed a pretty general deep learning architecture that in the future could be applied to other RNA-related tasks. For instance, it would be really nice to be able to predict RNA localization or RNA translational regulation or tissue-specific splicing patterns using this framework. I'd be more than happy to discuss these ideas with anyone interested. And finally, I'd just like to acknowledge David Kelly, the principal investigator with whom I've been pursuing this work over the past few years.